Okay, let's go ahead and get started. Good afternoon, everyone. My name is John Servos, and I'm the Director of Commercialization Education at Fast Forward Medical Innovation, also known as FFMI, and I'd like to welcome you to today's webinar, which is number 3 of our 6-part series with the Department of Internal Medicine. Before we get started, I do have a couple technical items that I would like to cover. If you'd like to ask a question today, please type it into the Q&A feature. Our staff will be using both the Q&A and the chat function to answer questions during the presentation, as well as share some additional links to resources that may be discussed during the 2 presentations that we're going to hear today. I'll note that the presentation is being recorded, and we are going to share a version of the recording and the slides with you, along with a post-program evaluation after the program via email. So watch your inbox for that. Today's webinar is titled Diagnostic Technologies, where we are going to discuss some of the unique considerations of biomarkers. And you'll hear Brad Martin, the director of FFMI, talk a little bit more about that here in a second. As I mentioned, the series is provided by FFMI, a unit of the Medical School's Office of Research. And today, in addition to the Department of Internal Medicine, we are providing this in partnership with the University of Michigan's Innovation Partnerships Group, which serves as the tech transfer office here on campus. Today's webinar, as I mentioned, is the third of 6 that we have planned as a part of this series. All offer some basic instruction on commercialization of certain technologies, and then a case study example by a faculty member who's working in that particular space. You'll see the February 1st date there as seminar number 3, which is today. Seminar 4 will discuss digital health technologies. 5 will be around biomedical devices. And then we'll wrap up the series on February 22nd with some next steps and some commercialization resources. So our 2 speakers today, you will hear first from Dr. Neehar Parikh, who is an assistant professor of internal medicine, also serves as the medical director of the multidisciplinary liver tumor clinic and the liver donor transplantation program. And then after we hear from Dr. Parikh, we'll hear from Jim Arthurs, who serves as a mentor in residence with the Innovation Partnerships Group here on campus and has a long career in small biotech in the biomarker and diagnostic space. So we are looking forward to hearing from both of them. Perhaps before we turn things over to Neehar, though, Brad, would you like to say a few things around the biomarker discussion?
Sure, just a quick comment. First of all, John, thanks to you and Catherine for putting this the seminar series together, and to our speakers today, Jim and Neehar, for their expertise and lending their valuable time. But just as a quick reflection on to how we got to this biomarker discussion, typically when we have run these courses previously for various departments or schools across the campus, we very often will have a seminar based on therapeutics, devices, diagnostics. And when we were talking about this program with your Associate Chair for Research, Viva, when we got to the topic of a diagnostic discussion, it became very apparent that in internal medicine, when we talk about diagnostics, or when she thinks about diagnostics, she very much wanted to focus on biomarkers. Specifically, if you're working in a disease area and your research leads you to a potential, what you think might be a biomarker, how do I know if this biomarker is truly specific to the disease state? How do I know when this biomarker may have value clinically, and how do I get approvals to use this biomarker clinically? That really became sort of the topic of discussion for diagnostics, i.e. biomarkers. And so we wanted to grab a couple of people here who had some expertise in how do you develop a biomarker project for a disease state? How do you validate that biomarker in an assay? How do you maybe measure that biomarker in a rapid, clinically relevant fashion? Those are the topics for today. And so I'll turn it over to our experts who can speak to this topic much more eloquently than I can. And again, I want to thank them both for being here. So John, I don't recall if Jim or Neehar is going first here.
Neehar plans to go first, and I've stopped sharing my screen, so you can go ahead and launch. There you go.
All right, thank you all. Everything looking okay here?
Yep, looks great.
Okay, great. Thanks, John and Brad, for inviting us, and Catherine for setting this up. Thank you all for attending. It's a privilege to be here today to talk to you all. Again, happy to take any questions after our presentations are over. I'm going to talk a little bit about the clinical perspective of biomarker validation and some of the roadmap from discovery to the clinic, and some kind of personal experience that we've had. I'll tell you a little story about how we've moved some biomarker work forward. So as mentioned, I'm a hepatologist, I'm a liver physician. I'm primarily interested in liver cancer, and we'll talk a little bit about diagnostics from that standpoint. These are my disclosures. So we'll talk about biomarker validation, why have a biomarker, some examples in liver cancer, and how do we get from bench to clinical use. We'll talk a little bit about liver cancer, present and future biomarkers. I won't talk too much about this. We'll talk a little bit about kind of the future and risk stratification biomarkers. But let's talk about the problem first. I think anytime you think about having a new technology or a biomarker, I think it's important to understand, what is the problem? What are you trying to improve on? So, right now there are national guidelines for liver cancer screening. Now, liver cancer is the 6th leading cause of cancer-related death in the U.S. It's increasing in frequency and incidence in the U.S. And it occurs primarily in 2 states, in patients who have liver cirrhosis, which is really just scar tissue in the liver and can be caused by lots of different things, or patients with chronic hepatitis B. And those are mostly Asian-born patients that have had it all their lives and are at risk of liver cancer. Right now, there are recommendations from our liver society called the American Association for the Study of Liver Disease. And the guidelines is ultrasound, which is an imaging-based test, every 6 months. So when you talk about cancer screening, this is a very intense type of cancer screening. Think about colonoscopy or breast cancer screening. A lot of these are either every 5 to 10 years, every couple years, but and sometimes even annually. But liver cancer screening is every 6 months. And we do use a biomarker in liver cancer screening. The updated guidelines are coming out. There's a blood test called alpha-fetoprotein that we use with the ultrasound to increase the sensitivity of ultrasound-based screening. We still need the ultrasound. We don't have purely biomarker approaches. And so this intense screening, an ultrasound is a separate appointment, requires patients to come in to get it done. So you can have these limitations. There's issues with false positives. People see things, they're not sure what they are. They end up getting MRIs, biopsies, et cetera. So lots of issues with ultrasound-based screening. And in this meta-analysis that was conducted, we looked at what's the heterogeneity in ultrasound performance. You can imagine it's provider dependent, right? So somebody comes in, does an ultrasound of somebody's liver, that may be different than it is from Ann Arbor and Muskegon. The ultrasound tech may be a different person who reads it may be a different person. So what we see is heterogeneity and sensitivity, which is a problem. When you're trying to detect cancer early, you want your test to be reliable across patients, across health systems. So this is one of the issues that we've seen with ultrasound, and really one of the problems we wanted to address. So we know that getting an ultrasound, as I mentioned, is hard. It requires a separate order, and what we see, nationally, compliance is really low. Compliance rates is around 10% for ultrasound-based screening in patients with liver disease, which is very poor. Even if we look at specialists, people who are getting seen by people like myself, gastroenterologists in the community, it only gets as high as 25% on average in this meta-analysis. So compliance is an issue. And, there are attractive, so this is one of the issues with ultrasound. And if you could have a point of care test, maybe you could improve compliance. And compliance really, it matters. Adherence matters. So this looks at test sensitivity and test adherence. So even if you have an inferior test, say if you have a 50% sensitive test, but if you can get adherence at least 50%, you could have a much more effective cancer screening strategy than if you have a very sensitive test that nobody uses. And so I think one of the attractive parts of using biomarkers in a clinical space, as opposed to imaging-based techniques, is that this paradigm is, the real world, is what we see. It's much easier for patients to get a point of care test, their blood draw at any lab, then come in and get an imaging like a mammogram, or come in and get a colonoscopy, et cetera. So this is one of the attractive pieces of biomarker-based screening. So I think I've touched on some of these advantages before. Ease for patients and providers. Maybe you can improve on sensitivity and specificity if you have the right biomarker. Can be more cost effective than imaging techniques, like other imaging techniques. Some people say, well, why don't everybody just get MRIs for their liver cancer screening, because the sensitivity isn't that great. But MRIs are expensive. We have capacity issues. And one of the key things that I haven't mentioned is that, you know, liver cancer screening is limited because we don't have level 1 evidence. There's not randomized controlled data in patients with cirrhosis. And so biomarker-based approaches, you know, if you can go forward with this, maybe we can generate higher levels of evidence behind its use and increase uptake. But there are disadvantages as well. You know, how do you deal with a false positive in biomarker-based work? We'll get into that when we talk about validation. Like, if you get a positive test, but then you get an MRI for diagnostic purposes, what does negative imaging mean? Does it mean that they're higher risk and they're to develop it? They may develop it, they may already have it, you just can't detect it, or is it really a false positive? And the other part is some of the pressures of bringing biomarkers to market. A lot of these are driven by biotech companies. And obviously there is this balance between academic validation and the interests of commercial entities in biomarker use. And so, you know, there's the potential for things coming to market before it's ready to use. And there's plenty of examples of this, but it's just something that we talk about frequently. So let's step back a minute. What is a biomarker? Really, a biomarker is any substance you can measure in the body. It could be blood, urine, saliva. Imaging is a biomarker. You know, a group here does this morphomics work with Grace Sue and Stuart Wang's lab. But this is, you know, these are examples of biomarkers, anything that's really measurable. I'll focus mostly on blood-based biomarkers. But then the question is, you know, how do you get from the lab into a human patient, here, somebody with a liver, which is everyone, but, you know, how do you get there? And there's actually several discrete steps for biomarker validation prior to clinical use. And these have been laid out by the NCI, National Cancer Institute, as a seminal paper by Pepe et al. And they really lay out the steps for adequate biomarker validation. I'll talk a little bit about them right now and give you some examples. So it's a 5-phase thing, and it's really akin to, you know, when we think about drug development. That's what this is really based upon. It's a way to validate things from a discovery phase into a clinical utilization phase, similar to drugs. And the timelines and costs can be on the same scale, to be honest. So there are several challenges with this, but we'll go through the phases briefly. So phase 1 is this preclinical exploratory phase. So this is how you discover a biomarker. It's exploratory studies, usually of cancer tissue or imaging, to identify potential biomarkers. And it must be reproducible. This early quality control is really important in order to say, can you develop an assay to measure this? Can you do it repeatedly, and can it be reliable? That's really critical to get any biomarker out of that initial phase. And so in this scenario, you're looking at patients with the cancer. Can you find, you know, proteomic signatures, genetic signatures, imaging-based signatures that you can measure reliably and reproduce in this early phase, preclinical phase? And you must be able to distinguish between patients with cancer and without cancer. These don't have to be huge studies, but there at least has to be a signal here. And again, I mentioned some of these microarrays, proteomics, to identify unique genetic and protein signatures. This is a very nice example, and I'll walk you through this. Is a very complicated slide. But this is a paper that was from the UCLA group that does a lot of work in liver biomarker development. This is extracellular vesicles. Okay, so extracellular vesicles are little, you know, compartmentalized, things that are, you know, basically vesicles that are able to be isolated in the blood. And there's a lot of interest in cancer because there's a belief that cancer cells excrete these, and you can distinguish them. So what they did was they had a chip technology, And they were able to purify these extracellular vesicles from patients with, cancer. And they were able to, have a technique where they could incubate these with antibody cocktails. These antibody cocktails were able to bind up these extracellular vesicles, and they were able to do basically a chemistry mediated capture using these antibodies that bound up. And so they had different targets. Again, some of these for those of you in the cancer world, like EpCAM, would be familiar, and they'd be able to bind up these extracellular vesicles. They were able to cleave them, and then eventually they were able to do these kind of RT-PCR analysis of these extracellular vesicles, and they were able to very well, using their bioinformatics core, distinguish between HCC and non-HCC. HCC is liver cancer, hepatocellular carcinoma, and so this is their discovery work. And they had some early validation data in here, kind of a phase 1, phase 2 type of study. But this is a really, really nice example of how you get from a patient with cancer and try to discover a potential biomarker and show that you could reliably distinguish cases and controls. Phase 2 is the next step. After you've done that phase 1, I've kind of alluded to this. And what phase 2 is patients with cancer versus patients without cancer. And this is really clinical assay development. Again, making sure that you basically can reliably distinguish between cases and controls. You can do there's consistency of the assay across laboratories, and you can really start the characterization if the biomarker is useful in early stage patients. So you want to know what the true positive rate is and the false positive rate. So this can really give you signals. Is this useful? You're also able to tell if there are other covariates that matter, depending on how big your validation set is. So age, sex, etiology of liver disease, degree of liver dysfunction are examples in liver cancer. But for whatever cancer, whatever disease state, what are the covariates that matter? And how do you stratify your populations? This is the stage where you want to try to think through those and make sure that this is going to be a biomarker that's applicable across the population of patients that are important to you and your disease state. Again, this is case control design, but the pitfalls is really finding appropriate controls, right? For example, in patients with liver cancer, a control patient is not a normal healthy patient, because those patients don't really develop liver cancer. That's not a screening population. In patients with liver cancer, it's patients who are at risk of development of liver cancer. You also want to make sure that those patients don't actually have cancer. So oftentimes we'll do make sure they have a 1-year follow-up. They haven't been diagnosed with cancer in that stage. We also want to make sure that they're early stage cases. I can very easily identify somebody with late stage cancer. That may not be a great outcome for that patient. What we really want to do is able to identify these patients in early stage. So you really want to have a mix. And I mentioned that ensuring controls are not actual cases. This is a very nice example of something called GALAD Score, which is 3 different biomarkers, age and sex, put into a formula. This is the initial validation study for that. And this is the type of things that you'll see. This is a big multinational validation of patients with HCC, which is liver cancer, chronic liver disease, from different countries across the world. They have, even in here, they have some other cancers like cholangiocarcinoma, which is a bile duct cancer, pancreatic cancers. I would argue the healthy controls is probably not relevant here, but this is what you would see. It's kind of cases versus controls. Now, the caveat with this phase 2 studies is you can overestimate your performance of your biomarker, because one of the things that dictates performance is prevalence of the cancer, prevalence of whatever disease. In case control studies, it's usually 1-to-1. You have a 50% prevalence or incidence. But in the real world, in liver cancer, patients have a 1 to 4% annual incidence. So once that incidence goes down, your performance of your biomarker may go down with it. So that's important to understand when you evaluate case control. And that's part of the reason that case control in itself is not adequate to getting into clinical studies. And honestly, this is where most biomarkers stall. And the reason is that there are not great phase 3 cohorts out there for many disease states. So what is a phase 3 cohort? A phase 3 cohort is a longitudinal cohort of patients who are at risk of the disease, and you collect their blood samples serially, and before their clinical diagnosis of whatever you're interested in this case, liver cancer. And then you're able to really get kind of real world incidence of the cancer. You're able to do serial testing of your marker over time and get a real sense of how your marker performs. Things like false positives and all that will be very, the performance of that marker in that setting will be, much more apparent. Usually what happens with these collections is, again, it's a serial collection that's prospective, and then there's a retrospective blinded evaluation of your biomarker. So you take all the samples that you've collected, over 6 months, and then you test your biomarker. And you don't know who has cancer and who doesn't. When you do the test, it's blinded. And then you evaluate how well does your biomarker work. And that's called probe design, okay? It's really to determine the ability of a biomarker as a function of time to detect preclinical disease. And then you can also define, you can set your thresholds for what a positive screening test would be based upon, false positives, true positive, those sort of performance metrics. So again, there's not a lot of these cohorts out there, but there's a lot of value in potentially collecting these cohorts so that you can test biomarkers and see which ones you want to move on to the next phase. And the next phase is a very resource-intensive phase. These are prospective cohort studies. Clinical utility trials is what we call them in the space. And what they are is a randomized study. Just like you would randomize for other studies, these are prospective cohort, which is where a biomarker is tested in real time with a diagnostic workup for a positive test. And you would test this against a gold standard. So in our case, we would test a biomarker versus ultrasound. And you would randomize patients into those. And you can stratify your randomization however it matters. The endpoints can vary. The endpoints could be stage migration. Can you detect patients at an earlier stage? A bigger endpoint would be overall survival. That's a harder endpoint and a bigger powered study. But it's important both for phase 3 and for phase 4 that you power your studies appropriately to what your incidence is. Okay, what your incidence of the outcome is going to be. Doing a well-powered study is really important in this case. Otherwise, your signal may not be, detectable. Again, this can determine the detection rate and the false positive rate in a defined population of patients. There's an alternate approach that many companies have gone, which is a more rapid. Okay, it's a basically they do the similar population. They do a gold standard test and a biomarker test, and they test each other at the same time. And the main outcome is performance of the biomarker for cancer detection versus the test. So in liver cancer, some companies what they're doing is they're doing a ultrasound, a biomarker, and then they also pay for an MRI. And so they get all 3 tests, and they can determine how well the biomarker performs against the ultrasound, with the MRIs being the gold standards. That's an example. What are the issues with this? The issues with this are false positive management. The longitudinal of the nature of this biomarker is lost. But you get rapid cohort maturation. So this is attractive from a company perspective. Phase 4 studies can take years, 4 to 8 years on average. There are several disadvantages. There's false positives, as I mentioned, the longitudinal performance of the biomarker. But an example is the Cologuard test, which some of you may be familiar with. It's a multi-targeted stool DNA test. This is a New England Journal paper. This is essentially the design that Exact Sciences, which is the company that developed that test, has used for this, and they're doing a study in liver cancer, and it's similar. In the phase 5 is mostly cancer control studies. These are generally kind of post-hoc analyses, and these are really addresses whether screening reduces the burden of cancer in the population. Ultimately, that's what we want. We want your biomarker to decrease the burden of cancer, these can be pragmatic studies where they're randomized to biomarker or the standard of care, or there can be kind of evaluation of the biomarker as in clinical practice, and it's decreasing cancer-related all-cause mortality. And again, there are several issues with real-world studies like this, like compliance, utilization, and underutilization of curative therapies. Again, just because you diagnose a cancer early doesn't mean it leads to a better outcome. You have to have a whole continuum of cancer care. Same thing with any disease. And the other issue is also overdiagnosis. And we've seen this in the prostate world, where PSA was overdiagnosing patients with indolent cancers, leading to lots of issues. So you have to, there's kind of a yin and a yang to early diagnosis. So the GALAD score, I'm going to just talk a minute about this. This is an example of something that we've worked with extensively. I mentioned it earlier. I showed the case control study. This is a composite score. These proteins have been around for 15, 20 years, and they just have never been validated in a way that allows us to move on from a phase 2 study to a phase 3 study to a phase 4 study. But that has recently changed. And again, this is a few biomarkers, age and sex. This is a nice example of a phase 2 study with 670 patients, 331 with liver cancer, and the AUCs for early detection were very good, very promising. And 86% early stage sensitivity and 75% early stage by treatment intent. So really excellent. But again, this is going to overestimate the performance. There was another validation that occurred that also showed this, but again, case control. And the sensitivity is the early stage we use a criteria called the Milan criteria. In these phase 2 trials was around 80% if you look at across studies. So really very, excellent. And so, how are we going to figure these things out? Again, these longitudinal cohorts, these phase 3 studies, it really is difficult to set these up though. You need infrastructure for serial collection with reliable follow-up. You have to make sure that your collection methodology is generalizable, and it takes time. We've been involved in this study from the NCI that has been serial collection in 1,500 patients with cirrhosis called the HEAD study. It's from the Early Detection Research Network. And it's taken, over 8 years for this cohort to mature. It takes forever for these phase 3 studies. And, we estimated an incidence of liver cancer in this population, but it was much lower because hepatitis C has been cured. And so that's one of the things that happens when you take a long time for a cohort to mature. Things happen. So I'm going to give you an example of a cohort study that was done here briefly from 2004 to 2006. Sample collection from patients with cirrhosis. So it was really like an early phase 3 study, one of the first ones that was done. They were serially collected for 6 months, serum and plasma. They were stored at -80, never defrosted or never thawed. And patients were followed with semi-annual surveillance until incident liver cancer, liver transplant, death, or loss to follow-up. And just last year, we used these samples that were sitting in the fridge for 15 years and did a phase 3 study looking at GALAD. And you can see here, GALAD, longitudinal GALAD, all that. Now, this is an underpowered study, but this gives you a signal of the AUC. Now, these AUCs are lower, again, these are phase 3, than in the phase 2 studies, and that's what you would expect. And sensitivity is around 50 to 60%, depending on the cohort. Now, this has been since replicated in a larger phase 3 study, that HEAD study that I mentioned from the EDRN, and similar performance measures. So, not 90% sensitivity, but pretty good for these, for biomarker performance. And again, if you can get adherence, that's great. Now, these are examples of national studies in liver disease that are currently ongoing. This HEAD study that I mentioned, 1,800 patients. The Texas HCC Consortium, going to have 5,000 patients. That's ongoing. So these are examples of phase 3 big efforts, lots of money, lots of funding that are necessary to move forward. This, we just got word of last week, is going to go forward. This is a national liver cancer screening trial. This is going to be funded by the NCI, the EDRN, using core funds. It's going to be a 15-site trial. We're going to be one of the lead sites, and it's patients with cirrhosis. And this is based upon all that work we did in the phase 3 space. This is going to move on to a phase 4 study. And this will start later this year. And so we've been hard at work putting it together. But it's patients with cirrhosis, well compensated, no other masses. It's going to be standard of care ultrasound plus AFP versus a biomarker. What biomarker? We're using GALAD. And the primary endpoint will be stage migration. This will be, if it meets its primary endpoint, it'll be moved on to a phase 5 study, which will look at HCC-related mortality and harms. And so this is a combined hybrid design, and we worked with the folks at Dana-Farber already, but this is actually happening, and we're super excited about it. And it's, there are 15 sites in the US that are going to be involved. So what are some logistical considerations when you're thinking about biomarker from a clinical perspective? You have to really understand your test performance for early stage detection. Your hazard here is that you could have an inferior test to imaging-based screening. Some solutions are non-inferiority design for acceptable margins, and a pragmatic trial design to incorporate the impact of adherence. As I mentioned, you can improve your adherence. Doesn't matter if your sensitivity might be marginally lower than those patients with standard of care screening. How do you manage false positives? You have to really, if you have a false positive, how are you going to manage that down the road? That's something you really have to think about. It's, your hazards or lack of care pathways for false positive results, and you really don't know what the implications of false positives are on the future cancer risk. Longitudinal trial design to understand and delineate the optimal care pathways really are important. So I think this is where these longitudinal studies are critical. Costs, lack of payer coverage. Jim will talk a little bit about this. Low liver cancer incidence will need high numbers to screen. So there really has to be rational price setting from a company perspective. I will say that having a company as a partner is really critical for any development of these. You need more funding than things like the NIH can give you to really run these trials well. And so these partnerships are really critical. And a lot of these biomarkers come from companies, and we're tasked with designing validation studies. And then you can also calibrate the intensity of screening based on patient risk. How do you do blood processing? Do you do central blood processing? Is there a lab? Can your lab run these? How do you do results across centers, and how do you make sure they're standardized? So there has to be quality control for shipping if you're going to do centralized, or you have to calibrate local labs for consistency. And then how do you report tests? Do you report tests back to the ordering patient? Do you do a level? Is it dichotomous, positive or negative? You have to develop these reporting pathways for results. This is really important with commercial partners. And then you have to, providing continuous test results really gives you the power of longitudinal testing. So this kind of Bayesian way of looking at tests, and if it's going up, even if it's not a positive test, that may be an early signal of cancer. And so these are logistical issues you really have to figure out as you develop these biomarkers. So biomarker development and validation is iterative process, but there are several good reasons why a biomarker-based approach could be advantageous for cancer screening or any disease screening. There are several promising biomarkers out there without sufficient validation. This is across disease states. And then, we're on the verge for liver cancer and other studies for large-scale clinical utility trials, which may change liver cancer screening and other types of cancer. And the NCI has a lot of interest in this approach. Thank you for your attention. These are my acknowledgements, my coordinator team, and some of the networks that we're part of. I'm happy to let Jim go from here.
Thanks, Neehar, and thanks, John, Brad, and Catherine for coordinating this. Thanks to everybody for participating. Let me get my screen shared here. All right, thanks. We're not stopping for questions right now, are we? We're going to go ahead?
Go ahead and go, Jim. Yeah.
All right, great. So I'm going to take a little bit different tack. I'm going to start from the point that says that you've done everything that Neehar went through in terms of basic research. By the way, he brought up a lot of points. There'll be overlap in some of the things that I might emphasize. But how do you get it from where he kind of left off in his talk to where somebody can actually use it in a clinic and bill for it and actually have it be paid for? So there are really 4 steps. The first one starts kind of with the validated biomarkers that were just discussed. However, if you're going to go into a clinical lab, there may be another biomarker validation or assay validation step in there. Whether you go to FDA or whether you're doing your own, you could face that. Then the lab itself, CLIA, FDA are involved in that. The basic process of handling samples, reporting them, and then billing. So what I intend to do is talk about some of the considerations in each of these 4 areas. But before we go there, what do you want to do? How do you commercialize? First of all, if you're at U of M, first thing you do is invention disclosure. And then the question always is, do we want this to go out to another company, or are we going to do a startup? By the way, those are not mutually exclusive. You could form a startup and continue to build value into your asset, and then exit to a larger company or sublicense to a larger company. So there are a lot of interesting things that you can do to improve the value of your technology and get it to market at the same time. And regardless of which way you intend to go or you're going, you basically have the same things that you have to do to attract that licensee or investors. You have to show that it works. You have to show there's a market for it. So where do you start with this? So I always like to say, what is it you're doing? What's the overall picture? So I think everybody's heard of intended use statements, probably seen them with FDA package inserts and things. So I always think to start at the beginning, what are you going to tell the market? And then what are you going to show them after you're done? So draft those as early in the process as you can. If you can talk about claims as well that you might want to talk about if you're going to go through an FDA clearance, claims will come into play that might affect your intended use statement. Customer discovery is primo in this area. Again, it's the old question. Is there a market for this that's worth investing in? And will it be accepted by the market? Is this an assay or a test that is usable and useful by clinicians? And I'm going to throw a pitch in here for quality systems. And I don't mean everybody run out and spend $60,000, $80,000 for a consultant to build you a quality system. Absolutely not. But start thinking of things like one component is a design history file where you're tracking changes in your assay, why you're making them, what data supports the changes. Because if you go toward an FDA clearance, this question will come up very early about show us your design history file. So also it's not a bad way to manage yourself. So as early as you can, just start thinking about things like that. Maybe get yourself familiar with the quality system contents in the FDA requirements and in the CLIA requirements, and pick the things that might help you as you're evolving. Even before you decide you're going to spin out of the university, there are things you can be doing, like the design history, like tracking lot numbers, like making sure all the equipment you're using and all the fridges and autoclaves and things you're using are qualified and measured and monitored. So things like that, if you get record of that while you're developing or validating your assay, it just adds credibility to your results in the end. And then really early on, engage people who can help. Fast forward medical innovation, FDA themselves, Innovation Partnerships is a great organization within U of M called MISHAR that has constant connection with FDA, and they can give you advice on things that you might be able to do early on to prepare for that. So why do I think quality systems are so important? I don't mean to belabor this, but you want to head yourself off from having any kind of out of control processes. If you don't pay attention to some of this stuff early on, it'll catch up to you when you talk to somebody about running your test through a CLIA lab, or if you're talking to the FDA. And clearly, the board and investors, employees will lose a lot of confidence if they don't see some control. I'm not speaking the subject of quality per se, although that becomes part of it. The quality system comes into play in terms of how you operate your company. I use the term internalize it within your company or within your operation, within your lab. Definitely phase it in. Work on function over form. I mentioned the design history file. There are examples of that. If you're keeping your design history notes in a notebook, that's the function over. You don't need to go follow somebody else's form. This is not at the time when you should just copy what somebody else does. You should implement things that are practical for you, for where you are, and where your assay is. So let's go to those 4 steps. Some considerations here. I'll go through these fairly briefly because any one of these subjects has a myriad of detail and a lot of interrelations. So just want to highlight some of the considerations, some things you got to think about. Obviously, your platform and any equipment that is involved will become important as you move through trying to raise money or trying to commercialize an assay. And anything you can do to build a spec sheet for this kind of thing will help. You talk about the kind of biomarkers, a single marker compared to one that's running with an algorithm. The algorithm itself, in my opinion, becomes a component of the assay and needs the same care and feeding as any other part of the assay. There's a lot of work now being done to deal with software as a medical device. You may fall into that if you have a multi-marker algorithm as part of your assay. So it's something to be concerned about. If all you're doing is doing a multi-marker panel and all you care about is the value of each marker, and that's all you're reporting, that's a different thing that will help you decide how to phase or phrase your results reports. Your algorithm is very important. It helps drive, obviously, the value in your test results. So that's actually, I was happy to hear Neehar mention prevalence of disease. One of the things a lot of developers forget to do is factor that in when they're evaluating their biostatistics in terms of number of samples they need and what the results really mean. So all of that comes into play here as well. Spec sheet should handle everything from the sample itself to collecting it, shipping it, handling it, processing it. So I think everybody knows this. Any medical test has to be run in a CLIA lab, a CLIA registered certified lab. Although that's a lie. If you have a wave test, that can be run outside and you can get a lot of wave tests over the counter. The test complexity is either waved, moderate, or high. The moderate and high tests are the ones that run in a lab, a medical lab. The complexity level is determined by 7 criteria based on how much med tech involvement is required, how complex the test itself is, whether it needs further evaluation. And once you go through those 7 steps, you get a score of, it used to be if it's less than 12, it's a medium complexity. If it's above 12, it's high. Virtually everything I've been involved in, and these have been cancer diagnostics, are high complexity. So anything that has any kind of detail or complexity in it, in terms of running the assay or the content of the assay, usually would end up in high. So there are 2 options, lab developed tests where only the lab who developed the test can run it. They can make no claims. Or if it's an FDA cleared test, it can be sold in kits and run anywhere. These are not mutually exclusive. You can start, if you want to build your own lab or work with a lab, you can do a lab developed test while and service that while you are preparing an FDA application for clearance. Manufacturing is a big deal, especially if you're going to FDA. That's one of the big things they look at is the manufacturing process. I'll throw in the quality system thing. By the time you get to this point, you will have a quality system either if you've started your own company or if you're having to talk with people to license your assay out. You'll becoming very, very familiar with quality system. Again, quality system comes in. This is where all these 3-letter terms, 4-letter terms come in. You might see these in different ways. ISO comes in real heavily when you're talking about FDA manufacturing. It's really important and nice to build a flow chart, just like anything else. Pictures work, pictures help. To go from the sample, what's the sample collection spec, and then refer to all the SOPs at every step in the flow chart, so that there's a reference there for employees. It's a good way to communicate it to somebody who's interested in investing. This can be done even now while you're still in the university and research mode, and probably should be if it isn't, because it can kind of build the framework for moving it into a lab or moving it to an out license. So billing is always is what you drive for. The cash only is becoming a lot more popular, it turns out. There's a lot of concierge labs forming up where they don't even make a claim to insurance and they only deal with cash for running tests. The normal thing though still is to submit a claim to your insurance company, which then would go to either Medicare or to that insurance company. They all have their own policy manuals, but basically everything is based on CMS fee schedules, Medicare fee schedules. If you're going to do an LDT, I would never, and we've thought about this, we tried to do this, but I would never recommend trying to do the billing yourself. It's very complicated, not so much in finding the CPT code that you might use or the Z code that you might use, but every state, it turns out, has their own insurance company. So if you're servicing multiple states, you end up having to deal with different things in every state, different. It's a real profession in itself. There are codes. People talk about getting their own CPT codes, getting their own Z codes. Z codes are a little bit newer. They tend to be applicable more to the DNA-based tests, and they're. I'm not going to say there's. Well, I'll say it's straightforward, but not necessarily easy to do. Getting your own CPT code is a little harder. It takes actually some actual commercial experience before you can do that. So what's the cost of taking a test from the lab or from the academic lab through to a full commercial usage? It could be as low as $10 million, but it's tens of millions. If you think in terms of a drug, getting a drug cleared and out, it's hundreds of millions. Diagnostics are generally tens of millions. If you're doing your own LDT, lab developed tests, it could be a little less, but you still have limits on not being able to make claims, and the test can only be run in your lab or the lab where it was developed in. So how do you get help to do this? There are a lot of great resources within the university and within the Ann Arbor area, some of which I've highlighted here. This isn't all of them. I apologize to anybody if I've left somebody off. I won't go through all these in real detail, other than to highlight that the MTRAC life sciences funding is a really great way to get money to help move things along internally. It's non-dilutive and is extremely valuable for technologies that are tending to eventually license or out license. FFMI has great programs for commercializing. one of the better ones that I've been involved in is called FastPace, which is kind of an I-Core shaped program, which helps you with customer discovery and helps you begin to build a pitch deck for your company. Ann Arbor Spark is great in terms of actually providing state and federal funds for entity formation and some other project type work. They have a boot camp as well. New Enterprise Forum is a local organization that does great work at helping groups do business planning and pitch decks. And yeah, the rest here you can read. I won't go into all the detail, but of course, innovation partnerships, I have to put in a pitch for that as well. One of the newest things there is Accelerate Blue Fund, which is an investment arm that lets innovation partnerships actually help some of these technologies move along who aren't quite ready for VC or angel funding and have outrun all of the non-dilutive funding and grant funding that might be available to them. Innovation Partnerships also puts out a pipeline report to 600 or so investor groups on a quarterly basis and have all kinds of other networking and other services available. And of course, the classic angel and VC investors are out there. Anybody in this panel, in this group at FFMI or Innovation Partnerships can help you connect with those kind of people. And there are others, like I said, this is not an exhaustive list. So that's kind of my rambling kind of approach to how to go from there or from here to there without getting into too much detail in any of it. As I said, there's a lot of detail involved in any of them, and they're all interrelated. But just remember to start with customer discovery and do your intended use statement and your results report, and that'll help set the charter for your team and for communicating with investors. And that's the end of my talk. Thank you. I appreciate the time.
Well, thank you very much, Jim. And I'll point out that in the chat feature, I was able to link to several of the resources that you were mentioning on your last slide.
Thank you.
And as a reminder, we will send a copy of the slides and the recording to everybody via email after this call. But we do have a few minutes. If somebody has a question for either Jim or Neehar, please feel free to unmute yourself and ask or submit by the Q&A function. No questions at this time? Well, if you do have a post-event question, please feel free to reply to that. Oh, we do have one question. All right, go ahead. Wenju.
Yes, my name is Wenjun Zhu from University of Michigan, Renal Division, Department of Internal Medicine. And thanks, Jim and Neehar, for the excellent presentations. I have a question. I have a biomarker and for kidney disease progression. We have patent on this biomarker. However, the assay has been, it's a commercial assay. Would that be very different in developing this biomarker differently than what you mentioned?
This, in terms of commercializing it, you mean?
Yes.
Yeah. So what it sounds to me like is if you've taken an existing marker and the one thing you can't protect is you can't protect a protein and a gene and things like that. I think we all know that. But what you can protect from an IP point of view is method of use. And so if you've got a method of use that is novel, then there is a possibility, I think, to do that. Does that get to your question?
Yes, I think so.
Neehar, you may have some comment on that too. Sorry.
that those proteins that we're using in this clinical trial is a good example. That those assays have been available for 15, 20 years.
Okay.
But the FDA just gave Roche a designation of combining those proteins for this specific indication for liver cancer. So, these are commercially available, but the intended usage is what got the approval and the
Great.
commercial.
Excellent. Thank you very much.
But it sounds like you may very well have to reach out to the current manufacturer of that assay just to understand their willingness to partner with you in some degree, or to take this research-based assay and turn it into a clinical assay.
Fantastic. Thank you.
Okay, we are very near the end of our time, so we'll go ahead and wrap things up. Again, if you have a question that comes up, feel free to reply to that email, and we can work to get that answered for you. I want to close by thanking Neehar and Jim again for your time and expertise this afternoon. We really do appreciate it. It was a fantastic session. And I want to wish everybody else that attended the call today, I want to thank you and then wish you a fantastic day.